So this is the last part of Chapter 14's lecture. We're going to take a look at some of the changes that we actually see in terms of this metabolism process. So for example, we're going to look at glycolysis occurring at elevated rates within tumor cells. And so if I take a look at GLUT1 and GLUT3, right, those are going to be upregulated in terms of glucose uptake. So if I think about this, right, I yield far less ATP than the complete oxidation to carbon dioxide that takes place under healthy cells with aerobic conditions. So a tumor cell has to consume much more glucose to be able to produce the same amount of ATP. And so glucose transporters and most of the glycolytic enzymes are actually overproduced in tumors. So compounds that inhibit hexokinase, glucose 6-phosphate dehydrogenase, or transketolase block ATP production by glycolysis, and those would then deprive the cancer cell of energy and kill it. The next thing we're going to look at are the fates of pyruvate. So once I generate pyruvate from glucose under hypoxic or anaerobic conditions, I can generate ethanol and carbon dioxide. So this is the fermentation to ethanol and yeast. Under aerobic conditions, I'm going to undergo the citric acid cycle. Right? And so I'm going to generate carbon dioxide in plants and animals and many microbial cells under aerobic conditions. And then lactate, right, also can be formed under hypoxic or anaerobic conditions. So this is in muscles, erythrocytes, and some other cells, as well as some microorganisms. So the idea here is, right, the generation of ATP without consuming oxygen or NAD+. So there's no net change in the oxidation states of the sugars. And this is the reduction of pyruvate to another product. So this is necessary because I need to regenerate NAD+ for further glycolysis under anaerobic conditions. And so this is one of the ways to produce food from beer or yogurt or soy sauce. These are all methods that require fermentation. So there are some animals that undergo lactic acid fermentation. So this is the reduction of pyruvate to lactate, and it's reversible. So during strenuous exercise, lactate builds up in the muscle. This is generally less than 1 minute. The acidification of muscle prevents its continuous strenuous work. And so lactate can be transported then to the liver and converted to glucose there. This requires some amount of recovery time, so high amounts of oxygen consumption to fuel gluconeogenesis, and then the restoration of muscle glycogen stores is also important here. So this is an example of fermentations. This is a conversion of pyruvate to lactate. Notice the delta G is negative 25.1 kilojoules per mole. You can also see here, right, the pathway of glucose to pyruvate and the cycling, say for example, of the NAD+ into glycolysis that is generated through fermentation. If we take a look at ethanol fermentation, we're going to notice that we use pyruvate decarboxylase to generate an acetaldehyde. And then we also use alcohol dehydrogenase to generate ethanol. And again, we're regenerating that NAD+, which is the primary driver for fermentation. If we compare glycolysis versus gluconeogenesis, glycolysis occurs mainly in the muscle and the brain, whereas gluconeogenesis occurs mainly in the liver. So these are opposing pathways that are thermodynamically favorable, but they operate in the opposite direction. So the end product of one is the starting compound of another. And these are reversible reactions found within that are used by both pathways. So irreversible reaction of glycolysis must be bypassed in gluconeogenesis, so highly thermodynamically favorable and then regulated. So I can use different enzymes in different pathways, and I differentially regulate in order to prevent a futile cycle. So converting pyruvate to phosphoenolpyruvate requires 2 energy-consuming steps. So the first step, pyruvate carboxylase gets converted to, pyruvate carboxylase converts pyruvate to oxaloacetate. So this is carboxylation using a biotin cofactor. This requires transport into the mitochondria via malate. And the second step, the phosphoenolpyruvate carboxykinase converts oxaloacetate to phosphoenolpyruvate. And this phosphorylation from GTP and decarboxylation also occurs in the mitochondria or cytosol depending upon the organism. So gluconeogenesis is expensive if you think about the energy costs associated with it. So for 2 pyruvate, I need 4 ATP and 2 GTP and 2 NADH in order to gain 1 glucose. This costs approximately, right, these values, but it's physiologically necessary because only the brain and the nervous system and red blood cells can generate ATP only from glucose. So when glycogen stores are depleted, you need to get glucose from somewhere. So during starvation or vigorous exercise, gluconeogenesis is necessary. Animals can produce glucose from sugars or proteins. So sugars can lead to pyruvate lactate or oxaloacetate. For proteins, these are from amino acids that can be converted to citric acid cycle intermediates or to glucogenic amino acids. And then animals cannot produce glucose from fatty acids. So this is the production of fatty acid degradation, which is acetyl-CoA. And can't have a net conversion of acetyl-CoA to oxaloacetate. So plants, yeast, and many bacteria can do this, which is why you can produce glucose from fatty acids. If you take a look at table 14-4, this shows you the glucogenic amino acids grouped by site of entry. And if we look at the pentose phosphate pathway, we can actually see here the oxidative phase versus the non-oxidative phase and the role of NADPH. So the main products of the pentose phosphate pathway are NADPH and ribose 5-phosphate. So NADPH is an electron donor. It's reductive biosynthesis of fatty acids and steroids and the repair of oxidative damage. So ribose 5-phosphate is a biosynthetic precursor of the nucleotide, so it's used in DNA and RNA synthesis or in the synthesis of some coenzymes. The oxidative phase generates NADPH and R5P. The non-oxidative phase regenerates the G6P from R5P. So this is used in tissues requiring more NADPH than R5P, so such as liver or adipose tissue. And NADPH is going to regulate the partitioning into glycolysis versus the pentose phosphate pathway. If there's a G6P dehydrogenase deficiency, this can be fatal in cases of high oxidative stress. So for example, certain drugs, herbicides, and some foods. There is resistance to malaria, though, due to high oxidative stress in red blood cells. So we can actually see here that reduced glutathione, GSH, protects the cell by destroying hydrogen peroxide and hydroxy free radicals. It's the regeneration of GSH from its oxidized form, GSSG, that requires the NADPH produced in the glucose 6-phosphate dehydrogenase reaction. And that's it for chapter 14. Thanks.